Good afternoon, everyone, and welcome to the Blood Transfusion Education Discussion Group. So my name is Selma, and I'm here today with my co-host Tasha, and will be your host for today. This is meeting number 51. So today's speaker is Nicole Thornton. She's the head of International Blood Group Reference Laboratory, the Red Cell Reference Department, and the National Frozen Blood Bank. Nicole also coordinates the compilation of maintenance of the International Rare Donor Panel and advises on rare donor availability worldwide. Nicole is an acknowledged expert in blood groups, antibody identification, and rare blood provision, and is particularly passionate about discovering new blood groups. So in today's session, Nicole will share the remarkable journey behind the discovery of the MAL blood group system, a breakthrough that resolves 50-year mystery surrounding the AnWj antigen and revolutionises transfusion medicine and care for rare patients.
Thank you very much for the invitation to come and talk about one of my favourite topics, is blood group discovery. I thought I'd do a little bit of just more of an intro into what we do and why we do it, and then I'll particularly talk about the MAL blood group system, which has been quite a journey for us at IBDRL. I wasn't sure how many people in the audience would know about the history of the International Blood Group Reference Laboratory, so I thought I'd start with that. And we have a very long history. The lab was established in 1946. And it used to be in London at the Lister Institute in Chelsea. Looks like a very grand building. And this is the one that we always see when IBDRL is referenced, but the lab wasn't actually in that building. We were out the back in an outbuilding, but we can still say we were part of the Lister Institute. Not me, I wasn't born. So the two main functions for IBDRL was to provide a centralised service for producing blood grouping reagents, and these were shipped around the world. And then the other main function was to provide a red cell reference service to what was then the newly formed UK National Blood Transfusion Service. And I really love these old historical photos, just showing how I imagine our quality department might have a heart attack if they saw a serology bench like that these days, with us all working on top of each other and all sorts going on there. But they got the job done, and they were the pioneers when it came to a reference serology. And for red cell reference, the lab undertook complex antibody investigations from the National Blood Service, but also from international reference labs from around the world. They also were tasked with maintaining the International Rare Donor Panel, which started in 1965, and we still look after that panel today. It was a centre for training doctors and scientists, and back then it was really sort of groundbreaking, the serological blood grouping methodology. And we worked in close collaboration with the MRC blood group unit, which is also famous for Race and Sanger used to be based there. So today, red cell references at Filton NHS BT Centre, and this is our serology lab, the photos here. It's a small lab. We don't have any fancy automation. It's all manual serological techniques, so we don't need loads of space for the serology. But this is just one arm of what we do. Our DNA work is done in other spaces around the Filton building. So our role is to be a red cell immunohematology tertiary reference lab. And we undertake complex investigations which involve red cell antigens and antibodies, which are mostly rare antibodies, and also blood group genetics. We also run the NHS BT Rare Donor Screening Programme. We compile and maintain the International Rare Donor Panel, and we also advise on rare donor availability worldwide. We take pride in the fact that we train in blood groups and immunohematology, something that we really enjoy. And another part that is very enjoyable is the R&D side of things. And that's really what I'm focusing on today. It's the blood group discovery work that we do, but we also support many other projects, and not just within NHSBT, but with colleagues around the world. And one of the perks of that, and also a responsibility, I would say, is that we publish novel, rare, and very clinically relevant findings in most cases. So I've just highlighted here the ones that are sort of pertinent to today, but it's just to also highlight that the R&D blood group discovery work and these publications that come out all start really with the complex cases that we are sent, patient cases and donor cases. So we're in that really lucky position, I would say, where the line is slightly blurred between diagnostics and R&D, and we need our R&D to be able to resolve these clinically relevant cases. So the types of cases that we have referred to us are samples, immunohematology problems that haven't been resolved by another reference lab. They've usually been through 2 labs before they get to us, a hospital transfusion lab, followed by a reference lab, usually a national reference lab. And then if it can't be solved, then it gets sent on to us. We receive antibody identification problems mostly, and this is complex mixtures of antibodies, but our main referrals are antibodies to high prevalence antigens. We also receive antibodies to low prevalence antigens, which can be also very clinically important, but we also see phenotype anomalies. It's like the Rh variants, things like that. And nowadays we see a lot of phenotype genotype discrepancies. And we do a lot of all the confirmation of rare donor work for NHSBT. And all of these things need to be done to have a resolution to a case to make informed clinical decisions. And in these 2 scenarios here, I've just highlighted them because it's in these scenarios where blood provision might be really quite difficult. And that's sort of why it goes hand in hand that Red Cell Reference investigates these cases, but also manages complex rare blood provision for other countries and within our own country. So I thought we'd have a look at where we stand at the moment when it comes to blood group systems. So I think most people know about the common ones, the ABO, RH, MNS, so on, but there are actually 47 blood group systems currently ratified, and that was as of June last year. There are 366 antigens within those systems, but there are also additional antigens, what we call the orphans. There are 27 of those, and they're antigens that are in collections or the high or low prevalence antigen series of the ISBT, and we don't know the molecular basis of those ones yet. We don't know what gene encodes them. And of course, they're the ones that we definitely want to work on to try and resolve, because if you don't know the gene underlying a blood group, you can't genotype for that blood group. So in total, we have 393 antigens. And if we I put in green here just how many there were back just in 2020. And within that short space of time, we've had 6 new blood group systems discovered, and 26 new antigens since then. So it's really booming at the moment, and I'll go through in a minute why that might be. So I'm often asked, well, how do you begin when it comes to discovering a blood group, and especially a blood group system? And as I highlighted just earlier, for us, it usually starts with a patient, a patient with an unresolved case, an unresolved antibody specificity, or a patient that has an antibody to an antigen of an unresolved genetic basis. So one of those orphan antigens that we know about, we can define the antibody, but we don't know the gene underlying their rare phenotype. So it's a nice a link to have. I think it's an important link when it comes to research and development, to always link that back to doing something better for our patients. Now, we do have some sets of rules to adhere to, and the International Society of Blood Transfusion's Working Party on Red Cell Immunogenetics and Blood Group Terminology set out the rules for what the criteria are for establishing a new blood group system. And you can, this is the website, you can go to the ISBT website and click on the Working Party page, and there's lots of information in there, including all the current known blood group alleles, the systems, the antigens within those systems. And then there's the criteria as well for, so this is the proof that we have to provide to establish a new blood group system. And I'll go through, as we go through the MAL journey, you'll see what we have to do. We go to the ends of the earth for these proof experiments. So the candidate gene is usually discovered really early on in our study. But we don't announce what the gene might be because it might not be the right gene. Proving that a gene is responsible for a new blood group system, it requires a massive amount of work. And it cannot be done without a huge collaboration with colleagues. We usually are collaborating within NHSBT, within the UK, and also other countries around the world. So these patients come from all over, and these samples come from all over. And we really have to work together to generate the required proof to prove that a gene is responsible for a blood group. It has to be solid and undeniable proof. And that's why we spend so much time making sure that we do have the right candidate, and then that we have proven that a gene encodes a molecules that we believe has reside where the antigens reside. And this is because the errors could have severe consequences. Blood group genotyping is completely reliant on this proof work being correct. If we get it wrong from the outset, then a genotype result could possibly predict the wrong phenotype. So I just wanted to take a little minute to remind ourselves about what the difference between a phenotype and a genotype is. And I know it sounds basic, but I think visually it's nice to remind ourselves what we're actually talking about when we use these two terms, because sometimes they're interchanged and they're not interchangeable. They're very different. And you'll see as we go along, I like to explain things in quite a visual way because it's how I remember complicated concepts. So I hope you find it useful too as we go along. It's not being patronizing. I just find visual things an easy way to learn. So when we're talking about a genotype, this is the genetic composition of a cell or an organism. And we inherit our genes as two copies of a gene, and what we call alleles, from our parents. And then the phenotype is the observable characteristics displayed by a cell or an organism. And this phenotype is influenced by genotype and environment, which I'm going to go into a little bit more detail, just to show why all this proof work for a new blood group system is so important. So genotype and phenotype are not the same. A phenotype can be predicted from genotype. What I mean by that is that a DNA test doesn't define a trait, it can only predict it. And that's really important to remember. And when we think about it in our testing terms, a blood group genotype is some form of genetic sequence or detection of a single nucleotide variation, or what we used to call polymorphism, now called what a SNP is now a SNV. The terminology changes every now and again, but yeah, so we call them SNVs now. But then when we look at the phenotype, this is a blood group phenotype. This is an antigen antibody interaction, which leads to detectable agglutination or some type of evidence of an antigen antibody binding. And we have lots of tests in our repertoire to be able to show a phenotype. So now for the next complication, and that's epigenetic modifications. So epigenetic modification is a process where certain chemical markers are added or removed from DNA, or to the proteins or the histones that help package DNA and this controls how genes are turned on or off. So these epigenetic modifications include, I guess the one that most of us would have heard of is DNA methylation. And a good example of this is X chromosome inactivation in females. Otherwise, they'd have double the amount of that protein than the males do. So yeah, it's really clever. The human body is very clever. There are also histone post-translation modifications, or PTMs, and non-coding RNAs, and especially we might have heard about the microRNAs. And they eventually turn certain genes on or off and regulate the transcription process. And all of this is done without changing the DNA sequence. So if we look at the word epigenetic, and I like to break down the word because it once we understand the meaning of the word, it helps us understand the description that it's trying to imply. And the word epi is a Greek word for outside of or around. So we're changing the transcription process is being changed, and this is done without changing the DNA sequence. So it's outside of the genetic code. It's epi. So I don't feign to understand this, but I'm absolutely, like, it back to front, but I absolutely am fascinated by it. And it really is the buzzword at the moment because we've come so far with understanding the genome. But there's another whole layer of complication for how we go from a genetic code to our phenotype, which is quite exciting. So I like to think of this in another way, a less scientific way. This is how I remember these things. So those of you who know me will know that I absolutely love cooking. And so I like to think about DNA as a cookbook. And I'm going to use my favorite recipe in this analogy, and that's my favorite chili con carne recipe as an example. So the recipes in the book are our genes, and in the cookbook, the recipes don't change generally, and our genes don't change generally. But the little notes that we make in the cookbook, the epigenetic markers, tell us how to modify the recipe due to environmental or external factors. And in this case, the external factor is my husband, who is an absolute lightweight when it comes to spicy food. So the modifications to this recipe don't change the original recipe. I have to modify the amount of chili, and I've written in my book that how much I have to modify it, and you can see he really can't deal with chili. And so those modifications, they to the recipe don't change the original recipe in the book, but they do change the outcome. And in this case, it's a very mild chili con carne, and my husband not sweating and complaining whilst eating dinner, which is always a bonus. So yeah, if you think about it, your little annotations to your recipes, that's epigenetic modifications due to external factors. And if we look at the many environmental or external factors that can influence which genes are active and to what extent, it really is, and I love this picture because it highlighted to me things that I wouldn't have thought of. We all know that certain things in the environment can have a detrimental effect or beneficial effects on our health. And this diagram is showing things that we don't always think, things like psychological state, things like that. And it all has an effect on our body and our health. And it can be an influencing factor when it comes to epigenetics. So one of my favorite examples of this is flamingos. And flamingos are a great example of epigenetic factors on phenotype. And flamingos are very well known for their vivid, bright pink color, but their color isn't actually inherited, which I didn't know until I looked into it. It's not an hereditary trait. They're actually born gray or white. They're really cute, the chicks. They get their pink color from their food. So they're literally the definition of you are what you eat. And the food that they eat is high in something called carotenoids. And as a flamingo dines on algae and brine shrimp, which they eat algae too, it's high. The algae it's high in carotenoids, and their body metabolizes the pigments, and that turns their feathers pink. So even their egg yolks can be pink. And during mating season, this is important because their color is a direct reflection of how good at foraging they are and how healthy they are. And they use this to attract a mate in a courtship ritual. And they apparently do quite an elaborate dance. You can see it on YouTube if you're interested. Obviously, I've done way too much research on flamingos, but they are quite fascinating. But my main point, and I do have a point, is that flamingos are famous for their bright pink color. And it's not a genetic trait, but it's become such an important element of maintaining that species' existence. It's factored into their mating rituals, which it just highlights the importance of epigenetics on a species' existence. So if we remember as we go along that genotype plus epigenetic factors equals phenotype, it's not just a case of genotype equals phenotype. And the male blood group system's a really good example of that, and I'll show you that in a moment. I thought it would be good to remind ourselves of the genetic bases of blood groups because, of course, it's not straightforward. There are different genetic bases or different genetic events that lead to a resultant blood group. A lot of them are single nucleotide polymorphisms or variations, but there are other ways of encoding a blood group antigen. This can include deletions. It might be one nucleotide, a whole exon, multiple exons, or a whole gene even. Insertions, intergenic exchanges where we end up with hybrid genes. We see that a lot in the MNS and RH systems. And then, of course, I mentioned some where we don't know the genetic basis. So most of the commonly clinically relevant antigens that we know about, that most people know about, are this type single nucleotide polymorphism, except ABO and D, which is unfortunate. They're very genetically complex and they're arguably the most important clinically. So blood group genotyping isn't new. We've been doing it in some form since the early 90s, and we've been doing it and comparing it to phenotype correlation for a long time, which is why basic blood group genotyping in most cases can be extremely accurate. And we have a ton of phenotype data that we've been able to compare to over a long period of time. We're really lucky in our field that we do have that. The difference now is the scale and speed and coverage of the genotyping methods available and the sheer volume of data that can be generated for analysis. So we can see from this graph, and I love this graph, it really, I think, shows the effect of technological advancements, especially genomics in the most recent years, shown by the boom in the discovery of new blood group systems. But behind every candidate gene is an enormous amount of proof work to ensure that that proposed genetic basis does actually result in the phenotypes that we're predicting. And we can see the power of genomics just by the boom there. And it's quite a steep incline, I would say. So yeah, it has really revolutionized the way in which we're able to discover new blood groups. Now we hear also the term big data used quite a lot these days, and the genetic data is one facet of this big data revolution. Genotyping now allows us to analyze beyond that pertinent SNP and look across the whole gene or probe for all SNPs reported to alter a phenotype. It generates a huge amount of data. And the speed at which the technology is developed is amazing. I like to use this excerpt from one of our IBGRL reports from only 2010. And it was a report on an unresolved antibody to high prevalence antigen. And it said, I think we shall have to call a halt on the investigation here as we cannot possibly sequence all of the blood group genes in the hope of finding a mutation. So only this short time ago, it seemed incredulous to consider doing such a thing, but we can do that now, and it's relatively easy, just in one test run. And it's not that long ago that we wouldn't have even thought of doing it. Would have had to use Sanger sequencing. It would have been really expensive. We had to go gene by gene, time consuming, and you weren't guaranteed an answer. You're still not guaranteed an answer, but it's a much quicker and accessible test now. So what was once crazy is now extreme capability in our testing repertoire. We're in a really exciting time when it comes to that side of things. So we now have the power to generate the masses of genetic data on an individual, and that leads to a very accurate prediction of phenotype. But as with all testing, there's still some known limitations, and that's highlighted to us in our most recent study, which led to the discovery of the male blood group system. So if I just cycle back to the whole point of us doing this type of thing, as I said, it's down to patients. That's at the front of our minds when we're doing this kind of work. And in the case of AnWj, or the male system, the cases that we're talking about were antibodies, patients that had an antibody to an antigen of unresolved genetic basis. So I'd like to introduce you to AnWj. We've known AnWj for a long time, since the 1970s. The name's complicated, or it's a complicated system, as you'll see in a moment, but it was always going to be complicated, I think, because even at the beginning the name was an issue, because the first antibody that was defined was called anti-Anton. There was another antibody called anti-WJ in a different patient, and it was later realized to be the same specificity. So it's been complicated from the beginning. So we now call it anti-AnWj, which is a bit of a mouthful, and that's probably why we didn't call the system AnWj. But the AnWj antigen has been in the 901 series of antigens, of high prevalence antigens of unknown genetic basis for a long time. The AnWj is present on red cells and epithelial tissues of greater than 99.9% of people across different populations. The antibody is rare. It's usually due to suppression, which may be transient and associated with hematological malignancies. Now, this is an added complication, which I'll explain in more detail in a moment, which really did thwart a lot of the work trying to define what the gene responsible for AnWj was. But there are very rare individuals who have an inherited form of AnWj negative phenotype. Of note, a very large family were published by my predecessor, Joyce Poole, and colleagues in the early 90s, and this was discovered in an Arab-Israeli family. Anti-AnWj is clinically significant, and it has been known to be clinically significant whether it's inherited or a suppression case. They have caused hemolytic transfusion reactions, but it's not significant when it comes to HDFN. There's never been a case of HDFN due to AnWj, and that fits in with what we know about the antigen because it's not on cord cells. It's a really strange one. It turns on, it's not on newborn cells, and it turns on, it's different in different newborns, but a little way, like days after they've been born. So that's another mechanism that we haven't worked out yet. And when I've said here that both the suppression cases and the inherited cases have clinically significant antibodies, I think in the literature where they've been defined as possible suppression cases, it may be that they were inherited, but we wouldn't have known back then. And we can't go back in time and test those samples and see if they had the mutation at the time. But as you go along, you'll see why it's important that we now know what the mutation is. So it had been long thought that AnWj was carried on CD44. Now CD44 is home to the Indian blood group system antigens, and it was thought that AnWj it at least required CD44 to be expressed. But more recently, it was published that a rare SNP in a gene called SMID1 was associated with AnWj. But in the paper, the mechanism of that wasn't very clear. There was no proof work that had been done. It was just the genetics. So the ISBT Working Party, Terminology Working Party, did not support ratification of a new blood group system based on that study. They requested more evidence was requested. So we were very fortunate to recently discover the genetic basis of the rare inherited form of the AnWj negative phenotype. And AnWj was moved to the newly ratified male blood group system earlier this year. We took a whole exome sequencing approach to find a candidate gene. And as I said, these things, the gene is often found early on, and it was quite early on that we found it, but the proof work was really, really challenging, not least due to that added complication of the AnWj negative phenotype of the suppression type. Yeah, it's been a hard old slog. We first started in IBDRL trying to look into AnWj 20 years ago. Obviously, we haven't been continuously working on it for 20 years, but as new technologies became available to us, we took different approaches and finally got there in the end. So as I mentioned, this suppression type of AnWj is, in fact, most AnWj negative individuals have a normal male gene, but suppression of expression of the protein. And we don't know what causes this. We only know that there is a suppression effect. It's been described in the literature in several cases where it's been shown to be transient. So with the corresponding development of anti-AnWj. So they lose their antigen, they concomitantly make the antibody, but it resolves later. And they get their antigen back and the antibody goes away. But the antibody has been described to be clinically significant in that suppression phase, which it really does sort of go against what we're used to thinking about these suppression, what we'd essentially call autoantibodies. With AnWj, it's not been so straightforward when it comes to that. And then the cause of this suppression type isn't well understood. It's usually found in patients with hematological malignancies, and this is suggestive of some type of epigenetic change, which I've discussed, and which is why I've discussed that before going into MAL and the complications of MAL. So I like to think of it as we have the blood group flamingo situation, if you like, and if we keep that in mind when we're talking about the suppression type of MAL, I think that will help as we go through what we've learned through the study. So initially, to determine the genetic background of AnWj, we had 11 cases where we categorized them into inherited or suppression according to various criteria. Those with other family members who also typed as AnWj negative were assumed to be inherited cases, and the original Anton propositus was also assumed to be inherited. So this was due to the long-standing nature of our AnWj negative phenotype. There was no transience, it didn't come back. And suppression cases were assumed on the basis of factors like a positive DAT, clinical history of autoantibody, and whether it was possible to absorb and elute anti-AnWj from cells, of their cells. The remaining cases with a negative DAT, no hints of AnWj expression or clinical history suggesting suppression, were also predicted to be due to inheritance. So we put them into the inherited category. And then we went on to, once we'd done that categorization, we then took our sequencing approach, where we used initially an exome sequencing of 2 cases, and then we went on to sequence further AnWj negative individuals and then on to our expression. So let's have a look at the whole exome sequencing. So a lot of people ask, why did you only do 2? Why didn't you just do the whole lot to begin with? It's whole exome sequencing is still really expensive. So we choose wisely when we do this WES approach. We decided that the original Anton propositus and one of the published AnWj negative family members from the Arab-Israeli family were both likely to be strong inherited prediction cases, and we started with them, and they didn't appear to be related. We first looked for CD44 mutations because it had been associated with AnWj before, and we didn't find any. We then looked at SMID1 because of that paper that had been published saying that it might be it might lead to the AnWj negative phenotype, and we couldn't confirm to see if we could confirm the previous findings in the Arab-Israeli patient. And only that patient, only patient 2 was homozygous for SMID1 mutation, and patient 1 didn't have any mutations in SMID1. So for us, we have 2 individuals that appear to be inherited AnWj negatives, and one has a homozygous mutation and the other one doesn't. So that to us means that SMID1 is not really a candidate for the gene that encodes AnWj. So on this basis, we went on to see if we could find another candidate for the loss of a high prevalence antigen. And we would expect this to be a very rare homozygous mutation shared by both patients in the same gene. And like a missense mutation or a nonsense mutation. We did this as part of our first round of filtering strategies with the sequencing data, and we didn't find any that were this type of mutation. So what we then do is we move to looking for deletions of exons or genes, and we do this by looking at the coverage of the sequencing. And we were, I would say, lucky. Well, we found that both samples shared the same large deletion in the MEL gene, and exons 3 and 4 were completely missing, which we didn't see in any other control samples. So this was extremely convincing, meant the gene was a really good candidate. But of course, we need to have a look at what MEL is. We didn't know anything about it at the time. So we had a look, and the gene is on chromosome 2 with 4 exons and encodes myelin and lymphocyte protein, MAL. This is an integral membrane tetraspan proteolipid. Importantly for us is that there is already strong evidence showing that MAL is on erythrocyte membranes, and it appears to act as a receptor for Clostridium perfringens epsilon toxin binding. So when you find a molecule that you're predicting may encode a blood group antigen, knowing that it's on red cells is a bonus if that work's already been done. And so this was a strong candidate for us. So knowing this, we went on to do some more sequencing of the gene in other samples, and we found the other 3 unrelated AnWj negative samples, which we had categorized as inherited, also had the exact same deletion as patient 1 and patient 2. And when we looked at family members, we saw that AnWj negative family members were homozygous for the deletion, but the AnWj positive family members that were siblings of patients were either and parents were either wild type or heterozygous for the deletion. 3 AnWj negative samples categorized as suppression were shown to not have any mutations in MAL. So this still fit with what we were predicting from our serology. Just on this picture here, there's some example NGS sequence, and it's showing a wild type control, a homozygous deletion with a big chunk missing, and you can see the ends of the sequencing reads before exon 3 and after the end of the gene. The bottom, which is highlighted in pink, is a heterozygous deletion with only about half the expected number of reads in the exon 3 and 4 region. So this is your heterozygotes. So it was really quite neat, and it fit in beautifully with what we were predicting from the serology. So we also checked CD44 and SMID1 in these additional AnWj negative patients, and no CD44 mutations were found in any of the samples, and only the Arab-Israeli cases had the SMID1 mutation, which I find this really interesting when we look, and it's a really good example. So let's have a look at it a bit closer. It really highlights the importance of having a diverse sample set when you're carrying out these studies. And the importance of proof experiments. So the paper that came before about SMID1, there had been no proof experiments done, and it was solely on based on samples that were from one population. So we found that the SMID1 mutation in AnWj negative members of the two Arab-Israeli families, they were homozygous for the SMID1 mutation as well as the male deletion. The heterozygous male deletion was also found in heterozygous when they were heterozygous for the SMID1 mutation in those family members that were AnWj positive, they were also heterozygous for the male deletion. But other samples, our unrelated and non-Arab Israeli samples, none of them had the SMID1 mutation. So if we look at SMID1 and male, they're located on the same chromosome 2, and SMID1 is on the other, just on the other side of the centromere from male. So what we believe is happening here is that there is linkage between SMID1 and male, and the male deletion and the SMID1 mutation, but only in the specific population, and that they're inherited together. But SMID1, we've shown from our other samples, isn't the gene responsible for AnWj expression. We went on to look at male expression in red cells, and this is where the proof experiments come in. And it breaks my heart that the proof experiments, we show you on 2 slides, the results of the proof experiments. This is years of work and years of failed experiments and things that didn't work to prove this. But these are the experiments that provided the proof that we needed. So when I say failed experiments, it's not that it didn't prove. It means it's not proven. It means that antibodies that didn't work, just conditions of experiments that weren't optimal, things like that. It takes a lot to optimize these experiments. So we found that AnWj positive red cells have male expressed on their membrane, which we could see by, so if you look on the first set of pictures is by serology. The next set is Western blotting, and then we showed it by flow cytometry as well. So you say, well, why'd you do it 3 ways? You should have just done it by the serology, but this is the type of proof that we need to have, needs to be undeniable. So we do it in a multi-attack situation so that it is undeniable. We also showed that AnWj negative red cells that didn't have the male deletion with the suppression type also didn't have male expressed. So this was really important to show that something else is going on with those suppression type, but it is male that is missing, that is making them AnWj negative. So it is really neat. We then went on to do some expression studies, and these are key really when you're talking about a new blood group molecule. And we did these in the human erythroid immortalized cell line, the Bella cell line. And we showed that our wild type Bella cells didn't actually have any endogenous expression of male or AnWj, which you might say, well, why not? They're human cell lines. And this isn't a surprise, really, given what we know about the situation with no AnWj expression at birth. It appears that these very young cells don't have the expression that we would expect. So what we're able to do is use the cell line as a vehicle, if you like, to do overexpression experiments. And we were able to show that expression of AnWj, it was there if we overexpressed the male protein, but if we overexpressed the truncated male deletion form, the construct, no AnWj expression was seen. So this is just undeniable proof, really, that you need male. Male is AnWj. And we did some green fluorescent protein tagging constructs, and it looked like, and this was quite interesting and surprising, it looked like the wild type male was localizing to the cell membrane, but the truncated male, the deletion, it was stuck inside the cell, so it's not even getting to the membrane, and that's why the antigen's missing, which is really quite neat. We also checked to see what happened. We didn't need to, but we wanted to have the full story, and because other things had been published about it, that we wanted to check to see what happened if we expressed male in the absence of CD44. So we did that in a CD44 knockout cell line, and we could still express male and AnWj normally, even if CD44 wasn't there, showing that we didn't seem to need CD44 for expression of AnWj. We did it the other way around as well. So they're very independent of each other. They don't need each other, as unlike what's in the literature and the prediction that they might. So after all of that, we have generated enough proof to go back and request establishment of a new blood group system. As I said, it's taken years, and often these things do, especially with such a complicated system such as AnWj, MAL and the AnWj antigen. But we were able to get all of the required evidence to be able to take it forward. And it was the MAL blood group system was ratified as the MAL. It's quite simple, actually. The system name is MAL, the system symbol is MAL, the number is system number 47, the gene name is MAL, and there's one antigen, and that's AnWj. And as I mentioned, AnWj is quite a mouthful, so we chose to, and we agreed on the name MAL for the blood group system, and the single antigen within the system is AnWj. So we get to hold on to all that history around AnWj. So what have we learned? I think I might be ending with some more questions rather than answers, but that's probably the fun of blood group discovery. What we know now is we can find rare individuals with the inherited type of AnWj negative phenotype, and we can do this by genotyping now, as long as you include it in whatever platform or whatever system you're genotyping with. What we can't do is predict the AnWj negative phenotype of the suppression type by genotyping. And I think this whole study really highlights the limitations of genotyping. It's not the silver bullet. It's not going to solve everything. It's just one piece of the puzzle when it comes to especially very rare phenotypes and complicated blood group problems. What it does do, though, even though we can't predict the suppression type, we've identified the carrier molecule. Now we've highlighted MAL, and it gives us the opportunity to study MAL. And this molecule is suppressed in individuals with hematological malignancies, but we don't know why. It's a chance now for us to really study it, or not me personally or us. It's other areas of medicine where MAL might be important, to see if we might be able to exploit this knowledge now for the use in diagnostics and drug therapies. So it's not just about transfusion when we do this blood group discovery work. It's about many other areas of medicine when we can give extra information about a molecule in the body. Also, red cells are really easily accessible compared to other types of tissues. So we now have a vehicle, if you like, to study MAL that's more accessible. As I mentioned, these studies are a huge collaboration, and this is some acknowledgements here of all those who took part in the AnWj MAL story and the study that has been going on for many years. It was a big achievement. Thank you. Happy to take questions.
Wow, that was brilliant. I think while we're waiting, I'll take the opportunity and ask my question, if that's okay. What are the challenges in detecting and managing anti-AnWj antibodies in transfusion medicine?
Yeah, so AnWj, well, anti-AnWj is the main challenge is it's an antibody to a high prevalence antigen, so it will invariably have to be referred to a reference lab for resolution. The antibody is known to be clinically significant, so it's not something, once we've identified it, we don't have to worry about. So we are very fortunate, and because I didn't have time today to touch on this, because that's another whole lecture in itself, is what we do have is a rare phenotype called the INLU phenotype. And And this is a Lutheran A negative, B negative phenotype, which is also AnWj negative. It's due to a dominant inheritance of another gene that isn't male, and it's not related to Lutheran either, that it essentially suppresses AnWj and other blood group system antigens, especially Lutheran. So we do have rare donors that are AnWj negative, not because of the male deletion, that we can use as a rare blood source. We're particularly lucky in England because we have we probably have one of the highest numbers of these donors in the world, and we have a bit of a hotspot. So there is available blood for those that require it. I think it's because of the literature and the cases that have been reported to have been clinically significant in suppression cases where maybe we don't have all the information to make that assumption. It's tricky. Because we know it can cause hemolytic transfusion reactions. We wouldn't want to transfuse over it if we don't need to.
Great, thanks so much. There's another question. Thank you, Kate. So thanks for this. Really enjoyable and accessible for non-scientists. Did all the research have a big implication on those that had confirmed AnWj? For example, did you have to ask the individuals to give a lot of samples over a long period of time?
No. What we were very fortunate, well, we're very economical when it comes to samples, and we're used to having to deal with small amounts. And we were able to use historical samples that were frozen to be able to extract DNA and do red cell experiments. There was only one more recent patient that provided samples especially, but all the rest were archive reference samples. So yeah, we were fortunate that we didn't have to do that.
That's great, thank you. Yeah, lots of thank yous, very interesting. A really interesting presentation. Well done to you, and thank you for all the hard work you and your team put into this. Thank you for this interesting yet complicated case. Can I just ask, is there any age restriction to your research on AnWj?
What's meant by age restrictions? Sorry, I don't understand the question.
Sandra, would you like to come in? I'll unmute you. If you put your hand up, Sandra, then I'll be able to find you and unmute you.
Oh, I see. Pediatrics. Yeah, no pediatrics involved. It was all adult samples that we studied. We haven't had any samples from children when it comes to AnWj, and that might be telling us something.
So it's clear that one of the biggest factors to the successful identification of this system has been resilience. What helped you to develop this trait?
Sorry, could you repeat that? I can't see it.
So one of the biggest factors to the successful identification of this system has been your resilience. What helped you to develop this trait?
Pig-headedness, maybe. I must say that a lot of people in our lab, I think maybe it's a prerequisite, is that we never say never and we never give up. We have some cases that we've been working on for a long time. I know older samples or patients that may not need the result themselves. When it's an inherited characteristic, their family members might, and other individuals might then benefit from this work. So again, it's knowing that we don't know everything and wanting the answers to those things, because it may have an impact on other patients, not just the patient we're investigating. Yeah, and curiosity.
Great, thanks. So since the MAL group is clinically significant, is there any close substitute group that can be given in situations where there are no MAL donors and there are no available MAL blood group in the blood bank?
Yeah, so that's the in-lieu phenotype that I mentioned. So they are essentially AnWj MAL negative, and we're very fortunate in this country that we do have donors.
Brilliant. I think that's all we have time for today. Tasha has kindly put in the link to the evaluation form. Would you be able to just share the links for the last slides, please? If there's any burning questions that we haven't answered, please email us at BTEDG, or I'm sure Nicole will be happy to answer your questions if we can't, and then we'll get in touch with her.